Okay, vielen Dank. Thank you very much. Since we were told that we have a bit of flexibility of time, I actually, given the occasion and the audience here, I would like to actually start from a few thoughts, general thoughts on the opportunities actually for European science in the current uncertain geopolitical situation. I have kind of a unique vantage point because I've been, in my short life, I had the opportunity to travel the world, and I've ended up with a very strange number of citizenships. So I'm Russian, Mexican, and Luxembourger. So I think, I don't know if I'm the only one in the world with these three citizenships. And actually, I almost got the German citizenship, but our application was actually, we never got a reply from the Ausländerbehörde for some reason, and so I almost have four. I've spent quite some time actually in Germany, almost 10 years. Now, the interesting thing is that most of my collaborators are actually Europeans, and many of them are Germans. But I actually did not meet them in Germany, which is very strange. I actually met them in the United States. In particular, many of them at the Institute for Pure and Applied Mathematics at the University of California, Los Angeles. So the United States is, I would say, it used to be the convention hall of the whole world, because that's where the largest conferences happen, the American Chemical Society, the American Physical Society, and also many institutes like the Kavli Institute, many mathematical institutes that gave the possibility for many people all over the world to come together and spend significant amount of time discussing things and coming up with breakthroughs. But this is no longer the case. For example, so I'm a European citizen, but I cannot travel to the United States. I will not go into details why, but it turns out that many of my colleagues are in the same situation. So I have heard from Max Planck directors and Nobel laureates who are European citizens and nevertheless cannot travel to the United States at the moment. So this is, I think, one can see this as a problem, but I actually see this as an opportunity for Europe, because I think Europe can now take over and become this beacon of hope of actually building institutions that allow all the people from all over the world to come here and actually get together for significant periods of time and do breakthroughs. And I think this can be enabled by analogs of Kavli institutes and so on. And I think Leopoldina should play a role in enabling such contributions. So, yes, thank you very much. I would like to now go to something I actually know about much more, which is science. So, let us travel back about 100 years ago. So, Erwin Schrödinger, almost 100 years ago, proposed his Schrödinger equation. And then, in a few years, I think he went a bit deeper to actually propose a dream of what we can do now that we have access to fundamental first principles equations that describe interactions between atoms and nuclei. In principle, since we have the Schrödinger equation, we can study the behavior of molecules, materials, biological systems fully from first principles. Now, this is really an amazing fact that still keeps me awake at night, that we have all the equations to understand all of physics, well, most of physics, all of chemistry, all of biology. And in fact, this is what Dirac famously said in his 1929 paper, that the equations are there, but solving those equations are so hard that we need to find approximations that actually enable us to push the applicability of those equations to increasingly larger systems and increasingly larger timescales. And that's what we have been doing for the past 100 years. But we are not there yet. We cannot simulate the whole cell from first principles of quantum mechanics. But we would like to do that. So today I will tell you how AI and machine learning have taken us and is taking us much closer to that goal. And I cannot lie to you, we are not there yet, but we have this dream, and we've come much, much closer, I think, today to the realization of the original Schrödinger's dream of understanding what is life starting from first principles of quantum mechanics. So Feynman told us that everything on Earth, almost everything, can be explained by the jiggling and wiggling of atoms. And so my talk is about the jiggling and wiggling of atoms and how we can scale the study of this jiggling and wiggling to systems which actually have biological relevance, starting from pure first principles. Nothing else but atoms and their positions. In order to do this, you need two fundamental principles, or two fundamental theories of physics. You need quantum mechanics on the one hand, and you need statistical mechanics that allows you to understand the dynamics of systems. And in fact, the developments of approximations to solving those complicated equations have yielded 2 Nobel Prizes. And so essentially, what we are trying to achieve with AI-enabled molecular simulations is to interpolate between these 2 famous Nobel Prizes, one given for quantum mechanical approximations, one given for statistical mechanics approximations. So on the left-hand side, we have Walter Kohn and John Pople. They've been awarded the Nobel Prize in Chemistry in 1998 for developments of approximations, approximate equations to solving exact quantum mechanics. This is called density functional theory and the so-called hierarchy of so-called post-Hartree-Fock methods to solve the Schrödinger equation. So these methods that they've developed are very accurate in principle. They can give you the ability to calculate forces that atoms feel in molecules. But they are only applicable to very small systems, tens or hundreds of atoms. On the right-hand side, we have the Nobel Prize given in 2013, again, Nobel Prize in Chemistry, given to Karplus, Warshel, and Leavitt for the development of so-called mechanistic approximations for force fields, of interatomic force fields that enable propagation, dynamical propagation of degrees of freedom for systems as large as biomolecules. And so these methods are very efficient computation. They take just microseconds for an atom. Nevertheless, these methods are not very transferable, they're not very accurate, and are not scalable from small to large systems. And so what we would like to achieve is really to interpolate. We want the accuracy of quantum mechanics, we want the efficiency of classical force fields, and we want to combine them. So this is what machine learning or AI in molecular simulations aims to achieve. So we wrote 2 review papers, which are shown in 2021, and in those papers we surveyed the state of the art of the field of molecular simulations enabled by machine learning. And so, as you can see on this picture, basically there is a very big breadth of systems, from small molecules, materials, biological systems, and big biological units. And in 2021, we could handle part of those systems. Small molecules, materials, which are dominated by local chemical bonds, but we could not access, for example, whole proteins in water or biological units. Just in 4 years, we've moved this arrow is from 2021, we've moved this arrow very far to the right, and I will report now in my talk what we have achieved in 4 years since 2021, enabled by AI. So let me go straight to what we can do today. So this, what I will describe, is a wide-ranging collaboration between my group at the University of Luxembourg, the group of Klaus-Robert Müller, whose talk you have seen earlier today. He is at TU Berlin and Google DeepMind, and also Oliver Ohnke is the leading author of this work. He is a research scientist in Google DeepMind in Berlin. So actually, this project started the first day of the COVID pandemic. I had a call with Klaus, and Klaus was starting a sabbatical in Google. So basically it would mean that we would have access to quite substantial computational resources that Google Research had, and also software engineers, which is something which you usually don't really have within our academic groups, okay? And so the question is, what we can achieve by this combination? Our scientific knowledge, AI knowledge, but also software engineering, data production, and quantum mechanical calculations at scale. So since pandemic hit, I was very interested to try to simulate the adhesion of the receptor binding domain of the SARS-CoV-2, right, of the spike protein, to the human cell receptors. Now, this is a very, very large system, so this was a very ambitious project. We did not really achieve the dynamics of that, but we are actually getting there today. And I will tell you what we achieved then and where we stand today. So the idea was the following. So let's, so biological systems, you start with proteins, right? You heard about AlphaFold. AlphaFold works for proteins. That was the initial point. And so we wanted to basically do a dynamical version, if you wish, of AlphaFold. So we wanted to simulate molecular dynamics of proteins. But proteins are all made of well-defined fragments. These are just amino acids, okay? And there are 20 natural amino acids. So basically the idea is, well, let us construct fragments of proteins which are made of amino acids, and we can construct it to a certain size, right? And then we construct a database. We run quantum mechanical calculations to generate data for forces that atoms feel in those amino acids, and then we can basically follow a divide and conquer approach. We make small fragments, we make large fragments, and then we put all this data in a machine learning architecture, so-called, for experts in the audience, equivariant graph neural network architecture. And we could then simulate, in principle, the dynamics of any protein system, including water, because we also have water in our data set. So we did this. This was quite a big endeavor, right? We had to calculate a few million fragments, and some of those fragments were actually very large, so we needed Google compute resources, we needed software engineering, we needed pipelines, and so this really was a very wide-ranging project, which was really enabled by this collaboration between universities and a big deep tech company. So let me show you what we have achieved meanwhile. So we have now this neural network that represents the forces that atoms feel in molecules, and we can apply it to many biomolecules. I will start with what I would call the fruit fly of protein folding. This is polyalanine in vacuum. Why are we doing this? Because there are experiments on the system, and if you cannot reproduce these experiments, you cannot reproduce any other more complex biomolecules. So let me show you again. So basically what we do is we start from an unfolded protein. It's called an extended, fully extended polypropylene structure, and we make it fold at room temperature, okay? So it folds in about 100 picoseconds, and it forms a helix. Now, so this is what should happen, and of course this is not a surprise. Now, a surprise is that this simulation takes about 20 minutes on a single GPU. I did exactly the same calculation in 2010 when I was a postdoc at the Fritz Haber Institute in Berlin using what we call explicit ab initio molecular dynamics. So you solve the Schrödinger equation for each configuration, you propagate it, and that took a year of compute time on 1,000 cores, okay? So we have accelerated the simulations by 7 orders of magnitude, 10 million times, in just 13 years. This is an amazing scientific progress. This happens very rarely in science, that you have such a huge acceleration, 7 orders of magnitude. And we can even do better. So now you can ask a question, well, we know that a polyalanine must form a helix. This is a well-known fact. If you put a polyalanine sequence into AlphaFold, AlphaFold will give you an alpha helix. So did we learn anything new? Well, it turns out that we learned many new things. We get full molecular dynamics trajectory as a function of time. And so now we can compare this trajectory with our method, which is called GEMS. This is what is shown on the top. So what we are showing here is the structure of the protein, which is measured by the number of hydrogen bonds that are formed in the structure as a function of time in the molecular dynamics trajectory. And so we start from this fully extended structure. The first thing we see is the formation of what we call a wavy structure. It's like an accordion. This was not known before. No one have observed these accordions. This accordion structure has no hydrogen bonds in it, but it's just an intermediate state that jumps into a helical structure. So this means that the folding process is not just local. It's completely global. So you preform a global accordion-like structure that then jumps into a helical form. So the folding trajectory is qualitatively different from what was known in the literature. But then, once this helix is formed, it starts alternating between two different helical structures, which is called alpha helix and 310 helix. It's well known from crystal structure dataset, the protein data bank that was probably mentioned in the AlphaFold talk, contains 310 and alpha helices, right? But this was never observed before, right? This was observed experimentally, but not in simulations, okay? So we can reproduce experimental results once again, and we can compare this to the state of the art, which is the Nobel Prize in 2013. These are so-called empirical force fields, which are widely used in the field. And if you look at the bottom plot, you will realize that the folding is completely different. There's no accordion-like structure, and you just form local hydrogen bonds, you form a rigid alpha helix, and you stay there. So we find two new insights, qualitative insights, which were not known before. The folding procedure, the folding process of the helix is different, and then there is dynamical alternation between different helical forms. So this means we go beyond AlphaFold. AlphaFold would not predict these two different structures, and we also go beyond the state of the art in empirical force fields in explaining the whole procedure. Once again, experimentally in water, not in vacuum, it's known that polyalanine actually alternates between alpha and 310 helices. So this just shows you, even for such simple protein folding problems, we obtain completely new insights by our quantum mechanical approach, and of course accelerated by 7 orders of magnitude via machine learning procedures. Now, why this happens? And this is actually a really interesting example of what I would call ab initio science versus empirical science. So empirical science proceeds by collecting experimental data. And if you look at the crystal structures in a PDB, crystallized proteins, alpha helices dominate. And 310 helices are very rare. So you can only use information about alpha helices because the 310 helices are not available in these crystallized proteins, because crystallized proteins is not proteins in our cells. They are much more flexible in our cells, okay? And so now we have an explanation of why this is the case. So basically what we can do is we can take this molecular dynamics trajectory that I just showed you, we can cluster it into 3 regions. You have the alpha helical region, the 310 region, and the transition region. And then you can build this violin diagrams, basically the distribution of energies of these different conformations in the different regions. And what you see is that if you do explicit ab initio calculations on the left, and then the machine learning calculations, you observe the same violin plots. They basically tell you that alpha and 310 helices have exactly the same stability because of entropic effects. But if you look at all existing force fields, which is what people use before machine learning, they make huge mistakes. They tell you that the green and the red distributions are separated by about 20 kilocalories per mole, which is a very large energy difference. Why this happens? Because they did not have any ab initio data for what a 310 helix is. They are biased to crystal structures which mostly contain alpha helices. So this is a bias of empirical science. And once you go away from empirical science and you start ab initio from the Schrödinger equation, you find qualitatively different pictures. Now, we don't want to stay folding small proteins. We want to have proteins in water, and we can also do that. Okay? So one example we did in this paper is so-called crambin protein. This is a 46 amino acid residue protein in explicit water. And we did full simulations, full molecular dynamics of this protein. And for example, you can see on the right-hand side below that we reproduce the solution NMR experiments, which gives you an ensemble of conformations of this protein in water. We reproduce it quite well, while again, the state-of-the-art empirical force fields give you very rigid structures that do not represent the experimental results. Another thing we can do is to go a bit deeper in this analysis, and we can actually analyze the molecular dynamics trajectory of crumbing in water. And what you see is that with a classical force field, or this empirical potential, you basically get a trajectory which looks like a random walk. So your protein starts from some equilibrium configuration, it jumps to a different configuration, and goes without any memory effect, right? So it just randomly jumps. While with a quantum mechanical force field, our GEMS approach, you get actually a continuous trajectory in this latent space, which tells you once again that the molecular dynamics is qualitatively different, and you get completely new insights into the dynamical fluctuations of these large biomolecular systems. So how do we go beyond? As I said, there's a Schrödinger's dream of simulating the whole cell from first principles. How do we go there? So we know actually chemically what cells are made of. So 70% of cells are water, and 30% are chemicals. Out of those chemicals, out of those 30%, about half are proteins. You have small molecules, you have RNA, about 6%, 2% are phospholipids, so membranes essentially, 1% is DNA, and then the rest, 2% is polysaccharides. So basically, we already have the dominant parts in our data sets. And so what we did after our work is actually working on what we call a quantum cell data set, where we construct all of the different structures which are missing, carbohydrates, lipids, nucleic acids, in order to have a complete representation of the biochemical space of these systems from first principles. So this is a lot of work. So constructing data sets is very, very hard. It's not as easy as just constructing structures and doing quantum mechanical calculations. There's a lot of work that goes into this, and this work is at all scales. It's chemistry, it's physics, it's computer science, it's software engineering, it's a lot of compute, and so back and forth. Okay? And so this should be not underappreciated. Constructing data sets is a very important undertaking which takes a lot of time and effort and so on. And so we've constructed in our group and also together with Klaus a range of different data sets that span this biomolecular space and also the space of materials. And I'm very excited also that we started recently working on materials and to extend this general purpose of foundation model actually also to material systems. Now, in order to have a true foundation model or a general purpose model, we need a few more ingredients. So first of all, we need to make sure that the data that we produce with approximate quantum mechanical methods is accurate enough. So we need to benchmark it to more and more accurate calculations. So we've been doing this in collaboration with several people to really check that the data that we produce has sufficient accuracy, what we call chemical accuracy of 1 kcal per mole for a wide range of systems throughout chemical space. The second thing you need is to include long-range interactions. So machine learning is not very good at describing long-range interactions because it usually learns local representations. For example, graph neural networks that were mentioned quite a lot here, they usually learn some local representation in an environment. But in physics, in quantum mechanics, interactions can be very, very long-range, and so we need explicit methods for long-range interactions, and that's something we've been working in my group for the past 15 years, actually. Once we have all these ingredients, we can put this together. And this is what we have done recently. So this paper in Journal of American Chemical Society appeared just a couple of weeks ago, which is again a collaboration with Klaus's group, and done by Adil, who is a PhD student in my group, and Torben, who is a PhD student, or was a PhD student in Klaus's group. And basically what we did is we collected all the different data sets that us and other people in the field have published. We've done explicit quantum mechanical calculations on all of these data sets using the FHI Ames code developed at the Fritz Haber Institute in Berlin, which gives us the convergence properties that we need for having accurate data. We then basically built an efficient machine learning architecture, which is an architecture actually developed by Torben, called Socrates architecture for SO3, Crates. So basically, which takes into account the angular momentum properties of local environments of atoms and molecules. And we've augmented this architecture with explicit models for long-range interactions. Once we have that, we get a technically complex model that we can then train on all of this data on about 20 million molecular fragments, and we get a model, if you wish, a foundation model or a general purpose model that can be applied to any molecular system, from a molecule in gas phase to interacting molecules to a biomolecular ensemble. It's efficient, so you can run simulations up to a few 100,000 atoms on a single GPU. So we did this, and we ran all the different simulations that, well, some of the different simulations that people are interested in. We've studied proteins, carbohydrates, lipids, nucleic acids in explicit water. For example, we've studied the lipid membrane in water, and we've looked at the different structural, spectroscopic, and electronic properties of the system and compared to experiments, and we demonstrated that a single model is able to semi-quantitatively describe all of these different properties. This was not possible up to now. So usually people develop technically, essentially, force fields that can only address a very small part of chemical space. And every time that you go out of that chemical space, you need to retrain your model. In our case, this is not the case. We have a single model, a single force field trained on all this large data and explicitly augmented with physics that can actually transfer between different chemical spaces. And this means we can study also interactions between proteins and DNA, proteins and lipid membranes, and so on and so forth. Now, of course, all this is in the future. So we've developed the model, and there is a lot of development that needs to be done. We are not finished yet. This is really the first model, and I'm sure it has many failure modes. It has to be analyzed in many different ways, and this is only really the beginning of the road. With this, brings me to the end. So I told you, or I've explained you what Schrödinger dreamt, and I highly recommend everyone to read Schrödinger's book on the physical aspects of cells. We've come, I think, in the last years, as I said, we've accelerated things by 7 orders of magnitude. This is not enough to simulate cells. To simulate cells, we need another 7 orders of magnitude. Okay? So I don't know how long it will take to do this, and actually it requires a lot of developments in statistical mechanics now to be able to do this. But it's an extremely exciting time, and there's so much to do in order to enable Schrödinger's dream, and I'm really happy for the younger people in the audience, because there is a lot of work for you to push all this methodology further. With this, thank you very much for your attention.